Personally, I consider Amy to be just like this exceptionally awesome human and also a really great mentor. And I hope you're all as excited as I am to hear what she has to say to us today. So a little bit about Dr. Trowbridge. She received her BS in integrative biology from the University of Illinois in Urbana-Champaign and has a PhD in ecology and evolutionary biology from the University of Colorado in Boulder. She's now a professor at the University of Wisconsin-Madison, and her lab focuses on research in forest entomology, chemical ecology, and plant-insect interactions. More specifically, her research questions center around identifying how various climate factors shift tree plant metabolisms and alter resistance to insect pests and other plant pathogens. Dr. Trowbridge's research is interdisciplinary, which means there's something in it for everyone, regardless of your specific research topics and backgrounds. She's also an excellent collaborator, which is what brought me to Amy last year. And we've been working together on some cool drought and tree defense stuff in Pinyons. And we're going to hear a bit more from her side today. So I could probably go on and on about how awesome Amy is and how awesome her lab is, but I'm going to stop here and let her work speak for itself. So here we have Dr. Trowbridge and her presentation titled Drought and Defense: What is the role of primary and secondary metabolites in driving pinyon pine mortality?
Okay, can you hear me okay? We're good? Okay, great. Gosh, thank you so much for that very nice introduction, Sneha. I'm probably blushing. And just for the invitation to come and speak in the seminar series today. I'm just so excited. And gosh, thanks, Rich and the Hofstadter Lab for hosting me virtually for this talk. And thank you all for being here and for just taking your time this evening to spend it with me. I hope the next time we can be in person. Okay, so today, the story that I'm going to really focus on is indeed about drought and defense. And I'm going to spend the next 45 minutes, hopefully just 45 minutes, talking about work that we've done to really understand how drought-induced production or constraint in the production of certain metabolites may lead to some trees becoming more susceptible to insect attack and ultimately to mortality. But first, let's see if this will work. My slide advancing? We're good? Okay, great. So first, I just want to acknowledge my amazing, amazing collaborators and also my funding sources without whom none of this work would be possible. The projects that I'm going to discuss today were supported by the National Science Foundation and the DOE. And I'd just like to specifically thank Henry Adams and Nate McDowell, who have been longtime collaborators on this work, and just they're just so fantastic to talk out ideas with, and are always enthusiastic about my crazy ideas, and are just basically my pinyon ecophysiology gurus. So this is in large part because of them. And then I also want to thank our current students and postdocs who are working on the Saviata Drought project that I'm going to briefly mention at the end of this talk today. I don't have any data quite yet. We've had some delays because of COVID, but they have just been working so tirelessly to make sure that we're going to have great high quality data from that. So just, so thank you and stay tuned. Okay, so as many of you are acutely aware, living where you do, global warming, together with these episodic droughts and heat waves, have been causing elevated levels of both chronic and acute stress that often leads to tree mortality across these large forested regions of the globe. And these drought-induced mortality events can have really important consequences, obviously, for ecosystem services, for the biosphere, biogeochemical processing, et cetera. And so while many of the pictures in my previous slide were actually taken from places around the Western US, some you may have recognized, these events are not just occurring in your backyard. So this is a map of tree mortality since 1970. And what you'll notice is significant mortality events on every continent except for Antarctica. And in this paper by Henrik Hartmann, which I really like, he notes that we still don't have a very good predictive framework to answer pressing questions about tree mortality. Questions like which species are going to be more sensitive to a given drought at different scales, which individuals within populations are going to be more vulnerable. And then one that really resonates with me is, can we actually quantify thresholds of physiological parameters and use those to better predict mortality under future climate change scenarios? So to address some of these questions, there's been this really big thrust of research to investigate mechanisms of drought-induced tree mortality. In other words, just trying to figure out why and how trees die. So a lot of groups have been specifically testing over the years whether trees die from hydraulic failure, whether they die from some sort of carbon limitation that can also lead into hydraulic failure. What is the role of biotic attacks? And then, is it really just an interaction of all three of these factors in some of these systems? And so over the years, a lot of really great work has focused specifically on these relationships between water and carbon. And because of that, the models have followed the data, where models of forest mortality mostly focus on tree physiological responses to drought, and rarely, if at all, on insect responses to tree stress physiology. Okay. So despite their important contribution to tree mortality in a lot of systems, not every system, destructive insects are rarely included in these models, like I said. And so really the research in our lab is trying to improve models by providing what I'm considering to be this kind of missing mechanistic link between climate, tree stress physiology, and insects through a better understanding of plant defenses. And specifically, we're really trying to understand insect interactions with host tree defenses that are being shaped by drought. So today I want to tell you a little bit about, what we've been piecing together through our research about how drought and herbivory interact to impact tree defenses, and then how those changes in tree defenses have cascading effects on plant insect interactions. And so just like a Christmas carol, we're going to kind of first visit the past, where I'm going to discuss some previous work that we've done looking at consequences of drought and herbivory on pinyon chemical defenses. And that research is really going to focus on this eruptive defoliator in pinyon pine. And then I'm going to discuss some more recent work where we've assessed the impacts of heat and drought on chemical defenses in pinyon. And that work is really going to have this eye towards understanding interactions with another eruptive species of bark beetle. And then finally, I'm going to just briefly mention how that work here in bullet point number 2 has led to our current drought project and what we're hoping to gain from this new design in terms of just identifying physiological thresholds of defense. Okay. So I'm going to start by telling you just a little bit about the interactive role of drought and herbivory on pinyon pine defenses. So when we think of conifer chemistry, or at least when I do, I usually think of the oleoresin, right? The resin is just such an obvious defensive component for trees. And one of the dominant secondary compounds in resin is a class of compounds known as monoterpenes. So these monoterpenes, some of which I've put here on this slide, are typically these cyclic 10 carbon molecules. They're produced by a pathway known as the MEP pathway that needs certain precursor molecules like pyruvate and glyceraldehyde 3-phosphate, or G3P, which comes from photosynthesis, to be synthesized. And so these compounds are primarily, they're found in conifer resin ducts, resin blisters, canals, etc., that are basically lined with these plastid enriched epithelial cells. So this is a cross section here, if you can see my pointer, of a conifer needle, and you can see these circles along the edges. The edges are these resin ducts, and they're lined, like I said, with these plastid enriched epithelial cells. And those cells actually synthesize the terpenoids, and then they secrete them here into this extracellular lumen space where they accumulate under pressure. And because these compounds have really high vapor pressures, they can also just volatilize through the cuticle into the atmosphere. And that's really what gives, conifer forests that great Christmassy smell. And so while the resin itself can serve as a physical defense, just flushing out organisms from the bark, entrapping or entombing them, they also can serve as a chemical defense and cause mortality because of their direct toxicity and antifungal properties. Let's see if I can advance this. There we go. Okay, so monoterpenes can be found, like I said, both in the tissues and also in the atmosphere, and in both states can influence a series of multitrophic interactions. And they can do so in one way in these what we call top-down effects, right? So it can impact the ability of an insect to locate its host plant for feeding. They can affect parasitism, so they can actually help parasitoids locate their hosts, their herbivore hosts. And then they can also influence insect oviposition and choice. They can also influence species interactions from the bottom up, where they can be directly toxic to specific insects. They can serve as feeding deterrents or stimulants, depending on whether the insect is a generalist or a specialist. And they also can influence an insect's immunity and growth. And I'm going to talk about this a little bit later. Okay, so that's how monoterpenes will impact plant-insect interactions, which is why they're ecologically relevant. But just from a physiological standpoint, there are lots of really cool and interacting factors that can influence the production, the synthesis of these monoterpenes in and of themselves. And so, like I said, there's lots of different factors that can influence monoterpenes. First and foremost, their synthesis is largely under genetic control. So, once we have those terpenes that are being synthesized, their emission rates are then dictated by various forms of resistance that can exist within the leaf tissues or whatever tissues are they're being produced in. We also know that herbivore feeding can induce the production of chemical compounds, including monoterpenes, by upregulating gene pathways. And just actually the act of feeding can expose the resin ducts to the atmosphere and also increased emission rates. And then finally, synthesis and emissions are influenced by a suite of abiotic stresses, included elevated temperature, elevated CO2, and also drought. So I'm going to focus today on herbivory and drought and their impact on monoterpene production and emissions in Pinyon. Okay, so many moons ago, I was really interested in understanding the impacts of seasonality and herbivory on monoterpene chemistry, and I wanted to know about it under natural conditions, and I was searching high and low for a good study system, and I was serendipitously told over beers about an outbreak of a tiger moth in the Pinyon juniper woodlands in South Central Colorado by a friend who just happened to be climbing there the previous weekend. And so this is where this love affair with Pinyon and all herbivores associated with it really starts, with this dry, hot American summer, a beer, and this eruptive herbivore, the Southwestern tiger moth. So when I started working in the system, I realized I knew nothing about it. And so I needed to learn some information just about like the natural history of this tiger moth, as well as just the temperature and precipitation patterns in the Southwest. So let's start with the tiger moth. So the Southwestern tiger moth is just this very cool native univoltine specialist. It feeds primarily on Pinyon. In typical years, you can see extensive feeding, usually in April and May, when temperatures are cool, precipitation is low. They then pupate in June, and there's obviously no herbivory, so no tissue damage during that month. Following pupation, the moths are going to emerge, and then they're in flight from about late July through mid-August, where then they find each other, they find their soulmates, and then they mate, and they look for suitable host plants on which to oviposit. And then the caterpillars will hatch from the eggs, and the larvae begin to feed gregariously during the early fall. And this is when they make these beautiful kind of silken white tents, which maybe you've seen. And they use these tents to undergo what's called an incomplete diapause, which allows them to just come out and feed a little bit when it's a beautiful winter day, but really most of their feeding and the bulk of the herbivory happens in the early spring. And so because you live in the system, you already know this, that temperatures increase throughout the summer, and that really the majority of the area's precipitation comes in the later part of the summer with the arrival of these monsoon rains. And so I was thinking, okay, there's a lot going on here, but how do all these factors influence monoterpenes and what might be the consequences for this herbivore? Okay, so we're going to start with emissions and then talk about what's going on in the needle. So what I'm showing you here on the y-axis are monoterpene emissions. And then you can see the month of the growing season down here on the x-axis. You can see that trees that were undamaged are in black bars, and then trees that had herbivore damage, at least 30% herbivore damage, in gray. And then the insects' general kind of life cycles here above the graph. And so what I want to show you first is here in May, during the peak feeding, you can see that these herbivore damaged trees are emitting monoterpenes at about 6 times the rate of undamaged trees. And I just want you to note here that we actually sampled branches that had no damage, but were on damaged trees. So this is really a systemic kind of upregulation and effect. And then you can see then that as the summer progressed, damaged and undamaged trees, they become no different in terms of their emissions. And then they kind of reach this lowest emission rate here in July at the peak of the summer. And what's interesting then is, if you can look here in August, is, once we have this release from seasonal drought stress with the onset of the monsoons, we saw that trees that were damaged earlier in the season actually emitted significantly more monoterpenes than those that were not. And, one thing to keep in mind is, this is when adults are searching for oviposition sites. So this may come into play when they're choosing where to lay their eggs, but this was not directly tested. So just something to kind of think about. Okay, so now this is what the concentrations look like in the needle tissues over the same time period. So you can see that there's this trend that herbivore damaged trees actually contained less total monoterpenes than their undamaged paired counterparts. And we think this is likely due because of those extremely high emission rates early in the season, and then that followed up by drought. And then what you can see is basically by September, we see no difference in tissue content between the two treatment groups. Okay, so what do we see in the system? Basically, what we observed is that herbivory and seasonality act in series to control monoterpene emissions and concentrations. So in terms of the emissions, we see high emissions in the early part of the season driven by herbivory. We see low emissions in the middle of the season driven by drought. And then with this release from drought stress, we observe this increase in emissions once again, but trees that had previously experienced herbivory earlier in the season actually emit higher levels of monoterpenes than undamaged trees. And so what's interesting to me here is that this is very much in opposition to what models actually think happens when they're simulating emissions and when we're doing kind of global estimates of VOCs, particularly terpenes. So these models just make this assumption that there's always this exponential increase in emissions as a function of temperature. So during the summer, what we would have expected is an increase in emissions. And in fact, in this system, we see some of the lowest emission rates. And so what this work really suggests is that in these semi-arid systems, we need to include drought, which seems to be the primary driver during the summer, and insects, which cause the increase in the spring and interact with that seasonality in the fall, if we really want to accurately model these emissions and their consequences, not only for insects, which of course I'm interested in, but also just for atmospheric processes. Monoterpenes can determine the oxidative capacity of the troposphere, and that has lots of climate impacts and consequences to think about as well. So, okay, so now I want to just go back for a second, because if you'll recall, these are the tissue concentrations. And here in May, you can see, these herbivore damaged trees tended to have lower levels of total monoterpenes, even though it wasn't significant, but there were significant differences in some specific monoterpenes. And so what we wanted to know was, might that influence the tiger moth's immunity or the outbreak itself? Right, so if it can influence their immune system and their immunity against parasitoids and maybe growth, it might actually exacerbate these outbreaks. And so I don't have time to go into everything, but just spoiler alert, the resulting chemistry does affect the herbivore's immune response. And so, like I said, I can't go into that whole experiment, but basically the monoterpene concentrations and composition that was characteristic of those herbivore damaged pinions resulted in this strengthened immune response in the tiger moth larvae. And that's likely because what we observed was they were able to eat more. They had higher consumption rates because assumingly the tissue was less toxic. And by being able to consume more, they were able to allocate more of their energy and resources towards that immune response. And so those two studies together, the field study and then working with the tiger moth immune system, really suggest this potential mechanism by which monoterpenes can contribute to positive feedbacks between herbivory, insect consumption, and the immune response during outbreak cycles of this pine folivore. So basically what was happening in the system is we noticed that, these larvae were feeding in the beginning of the season. It led to increases in emissions. And as a result, it led to lower concentrations in the tissues. And because these trees became then less toxic to the insect, they could actually consume more of it, and they could invest more of their resources into their immune systems and protect them from parasitoids, which then allowed them to survive and reproduce, thus supporting this outbreak. However, like all things, there are tons and tons of caveats and lots of other factors that could obviously alter this positive feedback that I'm suggesting in this system. One of which is just that the parasitoid host is using volatiles to locate the herbivore. And so perhaps those high emission rates are actually serving as real honest cues for them and actually brings in more parasitoids, leading to a higher parasitoid load or pressure. The other thing is that the immune response that we measure, this melanization response, it might not translate into resistance against parasitoids, especially in the field. But this is work that in my dreams, I hope to follow up on someday. Okay, so what I'm going to spend really probably the rest of our time together talking about is the second point, which is consequences of heat and drought on pinyon chemical defenses, thinking about bark beetles. So you've probably all seen this picture a million times, but anyway, I'm going to just give you my spiel. But basically, droughts they're not only becoming more frequent, they're also getting hotter. And these hotter droughts are also highly correlated with insect outbreaks. And the best example I have is from the early 2000s in the Southwest, where the region experienced one of the worst droughts on record. And so, like I said, this is a pretty well-known photograph by now, taken by Craig Allen in 2002. And he also took this great photo in 2004, just showing that the whole landscape in the PJ or in the pinyon juniper woodland really changed due to drought and bark beetles. And what's surprising to me, I guess, is that in light of events like this, we still don't actually know what tree traits make some individuals more susceptible than others to initial bark beetle attacks. And a question I have is, can we actually, if at all, quantify physiological thresholds that lead to increased susceptibility to destructive insects? So I think personally that knowing how monoterpenes and other defense compounds change in response to drought stress has a really incredible potential to just help our understanding of, bark beetle population dynamics, because monoterpenes in particular play crucial and diverse roles in bark beetle chemical ecology, including that of the pinyon engraver, Ips confusus, that attacks pinyon pine. And I say complicated roles because, high concentrations of monoterpenes can be toxic to bark beetles, while low concentrations can actually be to the beetle's benefit. And so I just want to give you a few examples here. So what I'm showing you here circled is cis-verbenol. And this is a minor constituent of the pheromone blend of the pinyon engraver. And it basically just aids in recruiting beetle friends with some other compounds. And it's produced basically through this hydroxylation of this negative enantiomer of alpha-pinene that's produced by the host tree. And interestingly, it's also the case for mountain pine beetle. They also make verbenol through the same process. Other monoterpenes like beta-myrcene, for example, they can serve as pheromone synergists for this kind of situation, but again, can also be toxic at high levels. So there's kind of this balancing act that's occurring in terms of dose-dependent responses. And so monoterpenes not only affect the beetles themselves, but they can also affect the fungal symbionts associated with bark beetles. So these are just some data from a paper by Wang et al., I think published last year, that just shows that the growth of Ophiostoma ips, which is a blue stain fungus vectored by bark beetles, was strongly influenced by 3 different monoterpenes that were induced, obviously not in Pinyon, but in a different species. And so what I just want to show you here is alpha-pinene tended to, that led to decreased growth, limonene and myrcene led to increased growth. And so I don't expect you to know what's happening here, but I just want you to know that monoterpenes not only affect the insects, the bark beetles themselves, but also can play a role in impacting the symbionts. Okay, so ultimately to make monoterpenes, it comes with a cost, and that cost is carbon, among some others. And so monoterpenes are synthesized through photosynthesis, but also from non-structural carbohydrates, both of which can become limiting under drought stress. And so, trees allocate carbon resources, these carbon resources, not only to constitutive and induced secondary metabolites or defenses, but also, to storage, to reproduction, and to growth. And so you can imagine, because terpenes are some of the most expensive metabolites plants produce by mass, there's likely trade-offs with these other pools and processes. And that just gets even more complicated when you start adding in herbivores and pathogens and then throw a drought on top of it, right? So how can we keep all of this straight? How to think about this? Well, our conceptual understanding of these trade-offs and processes under drought has historically been guided by the growth differentiation balance hypothesis, or the GDBH. And really, the idea is that because drought is going to impede growth, this is the growth line here, we see it dropping pretty rapidly, one can then expect an increase in non-structural carbohydrate availability, and thus maybe that can be allocated to defenses like monoterpenes, especially as drought kind of becomes more severe along this gradient. But, when we think about NSCs and drought, there's actually been quite a bit of conflicting literature about those dynamics. So some studies have shown NSCs to decrease during drought, some have shown them to increase, some have shown no change at all. And actually, a lot of studies that focus on NSCs and patterns of allocation tend to really focus on how it's getting allocated to below ground structures or storage, or how it regulates hydraulics. And very little thought has been given to how and when they might be needed for chemical defenses. So I hope I'm convincing you that secondary metabolites and monoterpenes are important, and they're not only important ecologically, but just important carbon sinks. And yet we have very little information about those links between drought, NSCs, and defenses, especially in mature conifers, and especially mature conifers in the field. So I had this really unique opportunity to evaluate physiology and defense in pinyon through moderate drought stress over time at the survival mortality experiment, or the SUMO experiment, at the Los Alamos National Lab, which was an experiment that was set up by Nate McDowell in about 2012. And so what that experiment really consisted of were these below canopy rainout structures that diverted about 45% of ambient precipitation away from trees. And then there were also these 16 OTCs, or open top chambers. This is Henry Adams. Some of you may know him. In this chamber here, and those chambers were, they were equipped with these like incredible heating and cooling systems that you would find like in a building. And that actually allowed us to maintain 4 different treatment groups. So we had trees that were under ambient conditions, trees that were just heated to about 4.8 degrees C above ambient, trees that were droughted, and then trees that experienced both heat and drought stress. And so they were taking a slew of measurements, and we came in and were able to collect needles and stems from 2012 to 2016, with a few gaps in the data because some babies were born during that time. Okay, so first, what questions can you ask using this design? There's so many, but of course, like I said, some of us were having babies, and so we couldn't do everything. So we had to just like prioritize. And so the first question that we thought to ask was just how does prolonged drought and heat stress, both just separately and combined, alter monoterpene concentrations in these different tissues? And then we said, okay, if they change, how do those shifts in both total monoterpenes and individual compounds, how do they relate to these different physiological factors? Thinking back to the growth differentiation balance hypothesis. Can photosynthesis explain them? Can growth explain them, et cetera? And then finally, we thought to ask what NSC pools, so sucrose, glucose, fructose, starch, might potentially be used or mobilized to support monoterpene production in the system. Okay. So now, I don't know why this, oh, there we go. It's like, why isn't that working? So we're going to talk about this one first, about the effects of prolonged heat and drought on monoterpene concentrations in these different tissues. So here I'm showing you total monoterpenes in the leaves, which is the top panel here, and then in the woody tissue, which is the bottom panel. And then these are trees in the ambient, the heat, the drought, and then the heat plus drought conditions. So what you can see basically is that in both cases, trees in this heat treatment had lower levels of total monoterpenes relative to both drought here in the black bars and also heat and drought trees. And you can also see a trend that heat actually may be exacerbating this drought effect here in the woody tissues, but that obviously was not the case here in the needles. And then compared to ambient, these data really say or suggest that heat alone may have like a very little effect or maybe even a negative effect on monoterpenes in some cases, but can act synergistically to enhance drought effects. And so I'm going to come back and talk a little bit more about individual compounds, but I want to stay on the total monoterpene train just for a little bit to not confuse you. Okay, so then how did shifts in monoterpene concentrations change in relation to like growth and photosynthesis? Well, first, I think it's probably useful to show you how growth and photosynthesis, how they responded to the treatments across time. And so basically what I'm showing you here are results from repeated measures correlation analyses, even though it just looks like confetti party, which I love. But every color line represents the relationship for a different tree that we tracked over the years. And then those correlations are used to calculate kind of this collated correlation metric that's shown here in each panel. So as expected on the top, you can see photosynthesis was reduced with increasing drought stress. So as these values, pre-dawn water potential gets more negative, that means the trees are getting more drought stressed, and you can see photosynthesis declines. And you can also see the same trend here when we look at primary shoot growth. So that's how the variables related to water potential and drought, but did they help explain our monoterpene data? And the short answer is no. Basically, all of the plant physiological variables that we looked at explain less than 12% of the variation in total monoterpenes for both tissues. So in the needles, total monoterpenes weren't correlated with any physiological variable, photosynthesis, conductance, growth, any of them. And that was the same for the woody tissues, except here where we did see an inverse correlation with primary shoot growth rate and total monoterpenes in the wood, which explained about 11%. Okay. And then finally, what NSC pools might be used to support monoterpene production. Okay. In the needles, so first I want to talk about the needles, and then I'm going to talk about the wood. So in the needles, we found that total monoterpenes, right here on the y-axis, exhibited this negative correlation with starch. And starch specifically that was measured at the same time in the same month as those monoterpene measurements. And actually monoterpenes in the needles weren't correlated with any other non-structural carbohydrate that we measured. Okay, that's the needles. In the wood, total monoterpenes in the woody tissues were negatively correlated with starch, but importantly, starch levels that were measured 1 month before we measured the monoterpenes in the wood. And you can see here that, those starch levels explained about 44% of the variation. And you can see here then, there's a relationship with total woody tissue monoterpenes and glucose and fructose, right? So the sum of glucose and fructose, also from the previous month, you see this positive correlation with total woody monoterpenes. And they explained, those two, glucose and fructose together, explained about 61% of the variation. So together, what these relationships suggest that's going on in the wood is that starch is likely hydrolyzed during drought to glucose, which is consistent with lots of other studies, but that those glucose monomers can then be used to support monoterpene production, which we observed being increased under heat and drought stress, and also we observed on a lag. There's kind of this 1 month lag. And so these particular NSCs are obviously part of the story, but there's variation that can't be explained by them, so it's not the full story. Okay. So I just want to come back here about heat and drought stress and the effects on individual monoterpenes, because we've been focusing on just total monoterpenes. And the individual compounds are just so important. So I want to give them some time here. So although we saw that total monoterpenes they were increasing under combined heat and drought stress, some key monoterpenes with potential roles in its confusus ecology actually decreased. And so these shifts, these kind of what seem to be subtle shifts, may actually make trees more favorable for bark beetles rather than well defended, which you might conclude if you just looked at the totals alone. So I just want to walk you through some of these. So for example, drought and combined heat stress, they tend to increase levels of both this positive alpha-pinene as well as its negative enantiomer, negative alpha-pinene, right? And remember, negative alpha-pinene is an important molecule because it's that precursor molecule to produce cisferbanol, which is part of its confusus's pheromone blend. In addition, however, our heat and drought treatments also decreased levels of other compounds, most notably this delta-3 carine. So the effects of delta-3 carine on Ips confusus have yet to be determined. We don't really know anything about it. But the negative effects of this particular compound on both aggressive and non-aggressive bark beetle fungi complexes suggests to me that its toxicity might be relatively common among taxa, and so we're considering that it could be. We're going to test it. But basically, given the benefit of this negative alpha-pinene as an aggregation pheromone precursor and the general toxicity of this delta-3 carine, the higher levels of negative alpha-pinene coupled with these lower levels of delta-3 carine may make these trees suitable for bark beetles instead of being really well defended, which you might think just looking at the totals. And so that is a possibility. We don't know for sure, but is something that we really want to look into more. Okay, and then just another line of evidence with this delta-3 carine and showing that it might play a role with this particular bark beetle and its ecology is, not only could delta-3 carine having it be in lower concentrations make it more favorable for bark beetles because it's less toxic, but it might also support bark beetle broods because of its effect on their fungal symbionts. So these are data from my PhD student, Mike Friedman, from some fungal assays where he had media amended with monoterpenes you would find in drought stress trees from Sumo and then in ambient trees from Sumo. And what you can see here is that the reduced levels of this delta-3 carine under drought actually seem to allow for growth that were no different from the controls, whereas higher levels actually seem to suppress growth relative to controls. So obviously, more assays are needed to understand kind of the synergistic and antagonistic effects of more complex mixtures, but it's kind of just an interesting result. Okay, so to summarize, what we found at SUMO is that drought seems to increase total monoterpenes, and it has variable effects on individual compounds, where some increase, some decrease, and some didn't change at all. We also saw that heat alone, basically we saw no change in total monoterpenes, and really no change in individual compounds, and the few that changed actually decreased. But when we looked at heat and drought together, we saw an increase in total monoterpenes that were inversely related to starch and positively correlated to glucose, suggesting that starch hydrolysis may actually help support the production of some of those monoterpenes. And not only did we see differences in concentration, but we also just noticed that there were differences in composition when we took a look at all these different individual compounds. And so the real question to ask next is, well, how do those shifts in concentrations and composition influence bark beetle choice and also the success of the bark beetle and their fungal symbionts? And obviously, we didn't kill any trees at SUMO, which as someone who studies tree mortality was a total bummer. And so then what we had to do is we needed to think of another way where we could actually identify thresholds of defenses under abiotic and biotic stress. So what I'm showing you here on the left is a figure from Nate McDowell's 2011 paper detailing this hypothesized situation where defenses need to decrease to a critical level along a drought continuum. And when they do, this will actually help support insect population growth and the impact of biotic agents on tree mortality. And so even though this has been hypothesized for a very long time, we still actually don't know at what point key defenses decrease when we're thinking about just these physiological stress gradients. And we also don't know how those shifts affect bark beetle choice and performance. And so, because these physiological and chemical thresholds are likely nonlinear, we need large gradients of drought stress that can allow us to observe as we surpass multiple thresholds. And so these are data from SUMO. These are total, twig monoterpenes as a function of pre-dawn water potential. And you can see here that we only managed to stress trees to about -3 megapascals, and pinyons usually hit their net assimilation point around here, about -2.3 megapascals. And so, there were only a few that kind of made it further down, and also we didn't kill any trees, so we couldn't really ask questions about mortality. Plus, we couldn't assess bark beetle choice or success because there were no bark beetles at the site. Apparently, when you put mature trees in a big chamber, the bark beetles don't want to go in there. And so that was a bummer, but not completely unexpected. So to fix that, what we did was we designed this new large-scale drought manipulation experiment in the hopes that actually, instead of us just making a lot of assumptions, we would let the bark beetles kind of tell us when trees are most susceptible and when they're most favorable to enter. So what we did was we started monitoring pinyons at the Sevieta National Wildlife Refuge in New Mexico back in 2019, which seems like eons ago because of the lost year. And then actually we managed in January of 2020 to establish a series of drought treatments that include a 45%, 75%, and 90% reduction in precipitation using these rainout shelters from ambient. And then we also have controls at the site, as well as a plot where a 45% treatment was established in 2010. And that plot happened to be sprayed, so the bark beetles did not attack it. And so while all the other plots died, that one survived. And so we've been actually able to track those trees over the, and really ask questions about acclimation, drought acclimation. And so for the past 3 years, we've been basically measuring at relatively high frequency, much to postdocs and students' dismay, lots of gas exchange, constitutive and induced secondary metabolites, not just monoterpenes, but all classes of terpenes, thinking about phenolics, lipids, amino acids. We're still measuring NSCs and from multiple organs. We monitor canopy browning regularly. We're collecting insects. We're looking at the entire arthropod community. We are tracking bark beetle attack rates, tree water content, mortality, you name it. And so the really exciting part about this project is that we've been able to track what we consider to be some critical plant traits through mortality in the field, which you'll be surprised how hard it is to actually kill a tree in the field using one of these drought experiments. And hopefully in that suite of traits we're measuring, this includes traits that correspond and are going to help us understand better bark beetle arrival and success. And so as you can see here, trees are definitely dying. This one down here, we measured in June of this year. It was about -7 megapascals in terms of its pre-dawn water potential. And that was in June. And by August, it was just toast and chock full of bark beetles. And so it's just been really cool to watch these trees over the past 3 years and to really kind of just be taking their vitals this whole time. And we're excited to see the data. Okay, so hopefully I have not gone too far over and in time, but also that I've convinced you that we really do need some ways to better predict when and where outbreaks are going to occur in the future. And up to this point, models haven't been that successful, mostly because they're failing to incorporate first principles of ecophysiology, secondary metabolism, as well as insect responses. And so really the goal of this research, especially with this new Abiotic project, is to really try and develop new models that can include these critical parameters. And so what I'm going to do is just kind of leave you with some take-home messages on this slide, and I will be very happy to answer any questions if we have any time. Thank you so much for your attention.
Thank you so much, Amy. That was great. We do have time. We have 10 minutes for questions. And we're also going to have a detailed discussion with the seminar class from 5:10 to 6 p.m. and the Zoom link for that is in the chat box now for those of you who don't have it. And yeah, whoever has questions, feel free to unmute yourself or type it in the chat box. And if you're in person, Connor can help mediate that.
Yeah, because if there's 10 minutes for questions, I must have been talking fast. So please ask me anything.
Hello there, Amy. This is Tom Kolb. I have a question.
Hi, Tom. Yes, please.
Yeah, so I really enjoyed your talk. I mean, a lot of it hits close to home to me, pinyon pine and drought and bark beetles.
Sure.
So, this is kind of a in the weeds question, but,
Oh, just how I like them, Tom.
Good, good. Let me get out my weed eater. So, it's about inferring, carbon use from relationships between NSCs and monoterpenes. And you showed us some interesting correlations, and I've done some similar work on ponderosa. We want to interpret those correlations as, this shows that the upregulation of terpene production is helping deplete, carbohydrates in the tree, and this might lead to carbon starvation. But I've had kickback from reviewers saying, this is far too loose. You really need to use isotopic labeling to pin this down. So my question is, are you using isotopic labeling, or do you know anyone who is, to address this kind of question?
Yeah, so first of all, I also get that pushback. So you are not alone. And I am not the one reviewing that, because I am not harsh on the correlation side of things. Because a lot of this is correlational, and especially doing this type of work out in the field. I mean, yes. So the short answer is we are doing isotope work, and we've actually done a little bit in pinyon using pinyon seedlings, but more so with ponderosa. and more looking at the volatiles than looking at the tissues, but we're starting to look at that now. It is so difficult to design a robust labeling experiment with mature trees in the field. I don't really know how to do that well. And so one of the things, or one of the pushbacks I would say with reviewers saying to use isotopic labeling is then you're really constrained to using seedlings and saplings, which of course takes away from the realism of a lot of what we're trying to understand. But, that being said, people have, drawn down starch concentrations, by putting trees in the dark, and then they label with, 13 CO2 for a few days and let the starch, for example, build back up, assumingly with this 13C label. And then you can, track that into these different compounds, especially if you have access to like, an IRMS, or you have a really skilled chemist who can help, if you're using GCMS, can help look at the spectra, because, there's a ton of fragmentation that happens when you use GCMS. And so actually counting all those carbons again and reconstructing that is more difficult than people understand. So I think labeling is a really powerful tool. And I think it's shown us a lot in terms of use for new and stored carbon, but it's really hard. And I just don't think, I just don't know how people can do this in the field, I guess is my concern. And I've thought about that. I've actually like started to write proposals about that. And I'm like, this is crazy. I was like, what are we gonna do? Dragging all these tanks out into the field, but definitely in seedlings, especially incorporation into monoterpenes or even isoprene or, MBO. I think isotope labeling is really useful. I think when you get into, tissue concentrations and there's a dilution effect, it gets a little tricky. So, but yes, we're doing it. It can be unnerving, but again, mostly on seedlings. I hope that answered your question and didn't make you feel really disheartened.
Well, I mean, you confirmed what our team concluded, that, yeah, I mean, this is kind of almost a flippant review comment. Oh, you should do this,
Yes.
You look at the practicalities of doing it, and it's not impossible, but it's formidable.
It's formidable. And it's like, I guess if an agency wants to give me $2 million to make it work, I'll make it work, but it is not easy. And yeah, so, well, good. I'm glad we're in the same boat. I'm happy to commiserate about reviewer comments on correlations any day.
Okay, thanks a lot.
You bet. Thanks for your question.
Go ahead. Tanera, were you going to say something? I had a
No, I was
question
just
as
gonna say we
well.
have time for a couple more questions. Go ahead, Rich.
I was wondering, you kind of treated the Southwest tiger moth separate from the Ipskin pieces.
Yeah.
And I was wondering if you saw any type of interaction going on with herbivory and bark beetle preference.
I didn't at that site, and I was only at that site for one season. And then I moved to a different site on the other side of the Continental Divide. But I am really, I'm really interested in, defoliators in particular, predisposing trees to destructive stem and wood boring beetles. We actually have been thinking about this, not in Pinyon, but in Doug fir, thinking about Western spruce budworm and Doug fir beetle. And just, we actually do tend to see a lot of overlap in terms of the defoliator and the presence of Doug fir beetle. And so one of our hypotheses is really that, just if you have this defoliator and it's really drawing, energy and resources and upregulating pathways for defense up in the canopy, that you're actually limiting and constraining resources that could go into defense in the bull. And so we've been thinking about that and did some preliminary studies this year with it. But actually, it was funny when I put this talk together, it was the first time I had really thought about that. I'm like, oh, I was like, maybe the tiger moth is predisposing them, but I had never noticed it. So I didn't think about it back then. But, you know, like with mistletoe and other defoliators that we see in pinyon, it is possible, I think, depending on the level of defoliation and severity.
Yeah, that's really interesting. My thoughts were kind of going at the potential difference in composition of the monoterpenes released. And
Yeah.
because you showed some effects from drought, it'd be interesting how that the herbivore would also interact with that chemistry.
Yeah, definitely. God, I wish I had like a lot of money and a lot of time. But yes, I think that is so cool. And I think a very real possibility. And again, it's like, I think it's so easy to focus on concentrations, but like you just said, I think composition is just so important. And even, you know, like I was just talking about some of these compounds that aren't, you know, a major constituent when you look at relative proportions really play such important roles. Like limonene, for example, we've known that for such a long time in such low concentrations, but yet such an incredible, you know, bioactivity. So yeah, I think, you know, composition really deserves a lot more credit than it's given a lot of times.
Yeah. All right. Thank you.
Yeah, thanks.
I have a quick question, Amy.
Sure.
So I was wondering if you looked at twig chemistry and bowl chemistry and if there was a difference in the two, or if you haven't explored that yet.
Yeah, so at Sumo we didn't. At the Savietta we have. And Shaylin, my PhD student, for those of you who don't know her, she has been really interested in looking at compositional differences in the needles, the twig, the bowl, and then even the xylem. And she has found, I wish I had this slide. It's just preliminary. I think it's only from maybe 1 month or 2 months, but what she's found is that composition is so different. Very much so. And so, you know, even though, you know, ips can, like pinyon ips can definitely, you know, colonize the branches, the upper branches of the canopy, and usually I find them up there. But, you know, we obviously also see them on the bowl. And so just thinking about those compositional differences and what they're exposed to, I think it's, now that we've looked at her data, because she was looking at monoterpenes, sesquiterpenes, and diterpenes, and the composition is just so different. And so what she's been thinking now is, you know, really sampling along the length of the bowl and into the branches to kind of get this vertical gradient, if you will. And we know NSCs vary along, you know, the axis, the vertical axis of the tree. And so it makes a lot of sense then that chemistry would do the same thing. But yeah, so I don't have that slide, but you and I can talk more about that if that's something you're interested in.
Absolutely. Yeah.
I think, yeah.
Follow up.
Yeah, definitely.
We have time for one more question. Okay, I'm assuming no one has questions, but we are going to have a discussion in about 10 minutes. So Amy, take a break and log on.
Just walk around my office for a little bit.
Yeah, I'm about to do that. I'm about to
do a lap. Thank you so much. And thank you everyone for listening and being here. I just really, yeah, I really appreciate your attention. I don't know who was here. I can't see anybody, but I assume there were people, so that's good. Okay, I think I have the link. So do you want me to sign off and then I'll reconnect in 10 minutes?
Yeah.
Okay, that sounds great. Thanks again, everyone.